So today uh, I will be discussing about another part that is given in your cell biology syllabus, that is your microbodies. So let's get started now. Well, microbodies, they are also membrane bound vesicles which are ubiquitous components of all eukaryotic cells except your mature mammalian erythrocytes. So these microbodies are membrane bound vesicles. They are also membrane bound vesicles which are ubiquitous components. Now, what does ubiquitous mean? That means they are found everywhere, right? They are the common components of all eukaryotic cells except in the mature mammalian erythrocytes. Mature mammalian erythrocytes don't have this type of microbodies. Generally, microbodies are spherical and their diameter is ranging from your zero point 0.3 to 1.5 micrometer. And these microbodies have a dense granular like appearance, granule like structure inside which granules are present, and the membrane that is bounded to these microbodies, they are about 7 nanometer in thickness. Okay, now cytochemically, if I say about the microbodies are cytochemically can be defined by their catalase activity. That means the enzyme that make up about 1/4 of all the proteins in the organelle. So 1/4 of the portion of these microbodies contain the enzymes having catalytic activity. The rest of the three part made up of some other things. These enzymes that is found within the microbodies, they protect the cell from potentially dangerous accumulation of hydrogen peroxide, H2O2, right? So what I have said that microbodies is composed of mainly enzymes, just like we have seen in lysosomes. Also in yesterday's lecture vacuoles, we have also seen microbodies are also made up of enzymes. About 1/4 portion of the microbodies are containing enzymes, and these enzymes are protecting the cell from potentially dangerous accumulation of hydrogen peroxide. They contain different enzymes, not only one type of enzymes, but they contain different enzymes which catalyzes specific type of oxidative reactions. Now, based on the enzymatic composition, microbodies are of two types, right? So, microbodies have different types of enzymes, and on the basis of the enzyme composition, microbodies are of two types. One is called peroxisomes, which are found on the leaf, which are found on the liver cells, and another one is glyoxysomes. Glyoxysomes. So, on the basis of enzymatic composition, microbodies are of two types. peroxisomes found on the leaves, found on the liver cells, and glyoxysomes, which are normally found only in the plants. I think so. Now, these organelles that we are calling as microbodies, they appear to arise from pre-existing microbodies. So, from pre-existing microbodies, these peroxisomes, these glycosomes are formed. Now, let us talk about the different types of microbodies. The first is your glyoxysome. Let us erase this whole thing. So, about the glyoxysomes, glyoxysomes are a type of microbody which occurs in the cells of yeast. They occur in the cells of yeast in neurospora and oil-rich seeds of higher plants. They also observe in the oil-rich seeds of higher plants. Glyoxysomes are absent in your mature and dry seeds. Okay, so these are the common things which are found. Glyoxysomes type of microbodies are found in the yeast, in neurospora, in oil-rich seeds, but absent in dry seeds. More about this function. Glyoxysomes was actually discovered by beavers in the year 1961 and also studied by Braden Batch Braden Batch in 1967, and they are very small spherical bodies which are found only in plants. Well, we can also find them in yeast, also in neurospora, right? And these glyoxysomes, they contain the enzymes for the glyoxylate pathway. That's why the name comes from. So these glyoxysomes mainly contain enzymes. They mainly contain the enzymes that are dedicated for the glyoxylate pathway, right? I think you know about the glyoxylate pathway. Glyoxylate pathway or the glyoxylate cycle, it is a variation of your TCA cycle or tricarboxylic acid cycle. Glyoxylate pathway is actually an anabolic pathway which occurs in plants, bacteria, protists, and fungi. And the main center of this glyoxylate pathway is conversion of your acetyl coenzyme A to succinate for the synthesis of your carbohydrate. So normally in glyoxylate pathway they are converting the acetyl coenzyme A into succinate. Okay, so that's why the name comes glyoxysomes, because they contain the enzymes for the glyoxylate pathway. Okay, and These, as I have mentioned, that it is found in the oil-rich seeds. They are found in abundance. That means high in quantity. They are found in the cells of germinating oil seeds and some fungi. The beta oxidation of fatty acid produce acetyl coenzyme A, which this acetyl coenzyme A will be further metabolized in the glyoxylate cycle to produce carbohydrate. And it also helps in conversion of stored lipids in germination oil seeds into glucose by a process called gluconeogenesis. So that means they contain the enzyme. Obviously, they contain the enzyme, right? They must contain that enzyme which helps to convert the stored lipid in germinating oil seeds to form glucose. So from stored lipid to glucose by the process of gluconeogenesis can be done by this here by the glyoxylase cycle with the help of the enzymes found in the glyoxysomes. Right. So this was about glyoxysomes. Glyoxysomes have a single membrane and their matrix is finely granular. It also has a core in the center. So glyoxysome has a single membrane and matrix is finely granular. The matrix of glyoxysomes is finely granular. Okay. Now glyoxysomes is somewhat more similar to peroxisomes. We will study next in this lecture. But the difference between peroxisomes and glyoxysomes is that they have a crystalloid core at the center. So if this is the glyoxysome and if I use a different color, this is the central dense particle core. So a central core is found on the glyoxysome and finally granules are also present, which I am representing in green color. Well, fine structures cannot be drawn with the help of this touch screen, but I am trying to draw it. So these are the finely granules and this empty portion is the matrix where the granules are present with a single membrane, right? And this core is about 6 nanomicrometer in diameter. Okay, 6.0, this is 6.0. Okay, so as I have said that these glyoxysomes contain number of enzymes like oxidase. It contain oxidase. It contain catalase, and other enzymes are also found that are not found in the animal cells. So they are found only in the plant cells. And this unique enzyme found in the glyoxysome is isocitrate lyase and malate synthetase. So two enzymes which are found in the glyoxysomes which are not found in the um, animal cells, the peroxisomes, is your isocitrate lyase, isocitrate, sorry for the spelling, isocitrate lyase and malate synthetase. These two enzymes are found within the glyoxysomes. Now, what is the function of isocitrate lyase? What is the function of malate synthetase? We have to see. Isocitrate lyase, also in short we write as ICL. This enzyme involved in the glyoxylate cycle, which catalyze the cleavage of isocitrate to succinate and glyoxylate. So the isocitrate will be cleaved by this ICL and form succinate with glyoxylate. Now this process even occur in the glyoxylate cycle, and together this isocitrate lyase with malate synthase, they bypass two decarboxylation steps of the tricarboxylic acid cycle that normally occurs in plants. So they are overcoming the steps of TCA cycle. Well, malate synthetase, what they do, they are also involved in your glyoxylate cycle pathway. Malate synthetase is an enzyme that catalyzes the chemical reaction of acetyl coenzyme A with water and glyoxylate to form malate and coenzyme A. So it catalyzes the condensation reaction. Malate synthetase catalyzes the condensation reaction and subsequent hydrolysis of your acetyl coenzyme A and glyoxylate to form malate and coenzyme A. So malate synthetase, what they do, they cause hydrolysis of acetyl coenzyme A and with glyoxylate and form malate and coenzyme A. So these two enzymes are important in the glyoxylate cycle, right? Now comes about the, let us see about the function of the glyoxysomes. So the functions of the glyoxysome is, one of the main function of the glyoxysome is to convert the stored lipids into sugars, as I have mentioned in this lecture. So glyoxysomes convert the stored lipid into hexose 2 sugars. So conversion of stored lipids into hexose, that means 6 carbon sugars, and this conversion of stored lipid into hexose sugar is known as gluconeogenesis. Gluconeogenesis. And this hexose sugar is utilized by the emerging seedling, which uses it as energy source and carbon source until it is able to produce their own sugar by photosynthesis. So all the what can I say, what I have said before, that The stored lipids which are found in the germinating oil seeds, they, with the help of gluconeogenesis, they are making the hexose sugar by the help of the enzymes found on the glyoxysomes, and now they will be using them as a source of carbon, as a source of energy, okay, during their germination process, and it will continue until they make their own sugars by photosynthesis process. The next function of glyoxysome is glyoxysome also function is in nitrogen fixation and photorespiration. So they involve in nitrogen fixation and in photorespiration. They also involve in your photorespiration. I think you know about the photorespiration process, right? Photorespiration means what? It is a respiratory process in many higher plants by which they take up the oxygen in the light and gives out some carbon dioxide, contrary to the general pattern of photosynthesis. So normally the main function of photorespiration is the dissipation of energy, where the stomata get closed during the daytime because of water stress. Photorespiration actually protects the plants from photooxidative damage by dissipating the excess of excitation of energy. So during the so for the photorespiration process, these glyoxysomes are also involved. Okay, and the third point or function of glyoxysome is the glyoxylate cycle. All the enzymes, most of the enzymes of the glyoxysomes involved in the glyoxylate cycle, which converts the acetyl coenzyme A to succinate for the synthesis of carbohydrate. So here the acetyl coenzyme A will be converted into succinate, which will be used further for the synthesis of your carbohydrates, right? So this was about the glyoxysomes. Now we will say about the peroxisomes. Okay. So about the peroxisomes peroxisomes occur in many animal cells and wide range of plant cells also. They are present in all photosynthetic cells of higher plants, protozoa, brown algae, fungi, liver oats, mosses, and ferns. Well, liver oats are what actually? Liver oats are mosses like type of plant. The liver oats that I am talking na, they are just like moss type of plant, mosses, that do not have a vascular system. Liver oat don't have a vascular system, and they they don't have the vein like tubes to conduct moisture and nutrients throughout the plants, and thus they are small in size. Okay, so peroxisomes, what I have said, they occur in wide range of plants, higher plants. In protozoa also they are also observed brown algae, fungi, liver oats, mosses, and ferns. Peroxisomes are also of two types. They are also of two types, and that those two types are liver peroxisomes and plant peroxisomes. So two type of peroxisomes are observed, liver and plant peroxisome. More to go about the peroxisomes. Peroxisomes are circular. They have a diameter between zero point 0.2 to 1.5 micrometer, and they have a membrane of lipid and protein molecules which enclose their granular matrix. Just like galactosomes, they also have a membrane. Peroxisomes also have a membrane bound which is bounded by lipid and protein molecules, and it encloses the granular matrix of the peroxisomes. In some cases, matrix also contain numerous thread or fibrils. Sometimes peroxisomes does not contain only granules, they also contain thread-like structure or fibrils, while in others they contain an amorphous nucleoid or dense inner core. Just like we have seen in glycosysomes, they have inner core. Peroxisomes may have a thread-like fibril-like structure, or they may contain an amorphous nucleoid or dense inner core, just like clumping inside. In many species, the matrix show a regular crystalloid structure. Okay, so I hope you have understood what I have said. So these peroxisomes was actually discovered by Rodin in the year 1954, and the detailing of the structure of peroxisomes was explained by Tolbert in the year 1969. Okay, since we know that these peroxisomes also contain enzymes, as the name suggests, peroxisomes, they contain the enzymes for peroxidase synthesis. So the peroxisomes contain the enzymes for the peroxidase synthesis. Okay. They are often called as peroxisomes. So normally these peroxisomes are found in close association with endoplasmic reticulum. They found in close association with mitochondria and chloroplast. Normally peroxisomes, as I have said, they contain enzymes for the peroxidase synthesis. In animals, they are found in the liver cells. They are found in the liver cells and kidney cells. Peroxisomes are found in the liver cells and kidney cells, and they are found in abundance of photosynthesizing cells. They are found in abundance of photosynthesizing cells. And in photosynthesizing cells, each cell may contain about 70 to 100 peroxisomes. Normally, peroxisomes develop from the endoplasmic reticulum, and the shape and size are variable, as I have discussed with you. The shape and size of peroxisome varies from 0.2 to 1.5 micrometer. It can be of variable size. Each peroxisome is covered by a single unit membrane made up of proteins and lipids. They have a matrix of fine granules. They have a matrix of fine granules like structure. They also have contain fibrous like structure as well. If you refer to any book or through the internet source, as I have said, that your peroxisomes may contain numerous thread like fibrils, or they may contain an amorphous dense new inner core like structure. Sometimes we can also see granular, most of the case we see granular structure in the matrix region of the peroxisomes. Okay, now let us discuss about the enzymes found in the peroxisome. Well, the enzymes that are found in the peroxisome are glycolic acid oxidase, gly okay, sorry for that, glycolic acid oxidase. It also contain peroxidase, which found in less quantity here. Catalase it contain mainly, largest. It also contain amino acid oxidase, amino acid oxidase. So these are the some common enzymes which are found within the peroxisomes. Okay. Normally in animal cells, peroxisomes mainly take part in oxidation of number of biochemicals, including it may also take part in lipid metabolism in the animal cells. It may in the animal cells it may take part in many biochemical metabolic process, right, including lipid metabolism. Now let us see about the peroxisomes which are found in the liver peroxisome that we are talking about. Okay. Now liver cells we call them as hepatocyte. A typical hepatocyte may contain about 1,000 peroxisomes. Now 1,000 peroxisomes may be observed in the hepatocyte, and they are found as in oval shape or spherical vesicles bound by a single membrane. So normally the peroxisomes which are found in the hepatocyte in the liver cells, they are in oval shape or spherical shape. Now the number of peroxisomes can be higher in fetal conditions or during postnatal growth, during liver regeneration and during liver regeneration. So when liver is regenerating, liver cells are regenerating, or during postnatal growth, or during fetal condition, number of peroxisomes can increase. Where postnatal growth means it has three phases: infancy, childhood, and puberty. During that time, the number of peroxisomes can be more than 1,000, right? Now, this number even larger, as I have said. Drugs such as aspirin and clofibrate can induce peroxisome proliferation. There are some drugs also known as aspirin. Well, you all know about aspirin, I think. Everybody are aware of aspirin. right? Which is actually aspirin, and clofibrate. These drugs can also induce the peroxisome proliferation. Peroxisomes contain a wide variety of enzymes, but the most common enzyme system which is found in the peroxisome is flavin oxidase catalase system. So the flavin oxidase catalase system of enzyme is commonly found within the peroxisomes. The peroxisome proteins are synthesized in the cytoplasm. Peroxisome proteins are synthesized in the cytoplasm, and once their protein synthesis or translation is completed, they are then imported into the organelle. So all the peroxisome proteins, the enzymes, are synthesized within the cytoplasm, and after the translation they are imported into the peroxisome organelle. So this was about the liver peroxisomes. Liver peroxisomes, what I have said, a typical liver cell or hepatocyte may contain about 1000 peroxisomes. It may be much larger also during the infancy stage. During the postnatal growth means after your birth, infancy, puberty, that time they may also contain large number of peroxisomes, or when liver cells are regenerating. Some drugs can also induce proliferation of peroxisome like aspirin and clofibrate. The main enzyme system that contain in the peroxisome is flavin oxidase catalase system. Generally, all the proteins of the peroxisomes, that is these enzymes, are synthesized within the cytoplasm by the process of translation, we know. And after translation, they are modified to give the rise of their secondary tertiary structure, and then they are imported to the organelle of peroxisome. In case of leaf peroxisome, if I say about the leaf peroxisome, the leaf peroxisomes are present in the photosynthetic cells of green plants. Leaf peroxisome are found in the photosynthetic cells of green plants. Especially they are found in the palisade cells. Especially they are found in the palisade cells of C3 plants. C3 plants leaves, and they are found in the bundle set of bundle set cells, better to say, of C4 plants. So normally palisade cells are the plant cells which are located on the leaves right below the epidermis and cuticle. So right below the epidermis and cuticle, the palisade seeds are found, and they are vertically elongated, a different shape from the mesophyll cells that lies just beneath them. Whereas the bundle set, as I have said here, the bundle set that I have said, bundle set is also found in the leaf. It is a, what can I say? It is a layer of compactly arranged parenchyma which surrounds the vasculature, and it conducts between the vasculature and your mesophyll cells. It conducts the flow of water from xylem to the mesophyll cells, and then to the intercellular spaces. Well, you all know about the C3 plants and C4 plants. It's depending on the pathway which they follow actually, right? Well, the C3 plants and the C4 plants normally, what can I say? C3 plants, they use the C3 pathway and they require cool and wet environmental conditions, right? So, we are talking about the leaf peroxisomes. They are found in photosynthetic cells, especially in the palisade cells of C3 plants and bundle set of C4 plants. C4 plant leaves, better to say, the leaves of C4 plants. They develop at the same time as plastids, and indeed, the two organelles often are near to each other in the cell. So, these leaf peroxisome are close to the plastid. Mainly, the function of leaf peroxisome is involved in photorespiration, obviously. So, they involve in photorespiration, right? Which is a light stimulated production of CO2, and photorespiration reduces the efficiency of the process of photosynthesis, right? Photorespiration, you can say them as wasteful process for the plant cell, because during photorespiration what we do actually photorespiration, as I have said, that they take up the oxygen in the light and gives out some carbon dioxide in simple process, right? And why we do photorespiration, we dissipate the energy where the stomata get closed during the daytime because of water stress. Actually, photorespiration protects the plants from photooxidative damage by dissipating the excess of excitation of energy, right? So it is a so the main peroxidome function in the photorespiration process, right? So this was all about your peroxisomes and glyoxysomes. I hope you have understood the whole concept of peroxisomes and glyoxysomes. Well, that's all for today's lecture. Okay, so please study the lecture, watch the video lecture, because I'm giving in YouTube. You can watch it multiple times so that you can understand the concept much clearly. And then for the notes, you can refer to the Verma Agarwal book, or from the Wikipedia, you can also watch, or from the biology discussion. If you search in the website, write the name of the organelle which you want to study and write biology discussion, then also you can get information, more information about that. So that's all for today's lecture.